Phase contrast microscope. Who discovered phase contrast microscope is by Fritz Zernike, is a Dutch physicist and got Nobel Prize in 1953. It helps to observe the living cells, that's the main things. And what does it do? It explores the fine differences in the refractive indices of water and cytoplasmic components of living cells to produce a contrasting image. This will explain in the coming slides. So this is the contrast microscope, phase contrast microscope, and the image obtained is like this. So there is a darker region and a brighter region. Two contrasting regions are the dark and bright. Hence it is called a phase contrast microscope. So the image obtained will be in the dark and bright contrast only. So in the bright field microscope, the compound microscope, you have to stain it. The stain is toxic to the organism or the cells and lethal to the organism or might have killed the organism. But in phase contrast microscope, no stain is required. The transparent object can be viewed, and this is based on the light intensity variation. So in the phase contrast microscope, when the light passes through the object, so this is a paramecium which contain various cell organelles, macronucleus, contractile vacuoles, and various organelles other. When the light passes through it, there will be phase changes. There will be deviation in the light rays. So this deviation in the light rays is made into a visible forms which can be detected by the human eye. That is what phase contrast microscope is doing. And what is the principle? Let's look at the principle here. A small phase changes in the light rays induced by the differences in thickness and refractive index of the different parts of an object. It can be transformed into differences in brightness or light intensity. This can be explained by this image. So suppose this is a cell. You can see contractile vacuole, granules, and nucleus is there. These differ in their thickness. They have differences in their thickness, and thus the refractive index. Refractive index means is the density, the different parts of an object. So they have low refractive index, high refractive index, and moderate. Contractile vacuoles low, granules high refractive index, and the nucleus moderate refractive index. And what happens to them? So when the light passes through the contractile vacuole, because of the low refractive index, there is little deviation or no deviation at all. The light passes straight. In the second picture, the granule shows high refractive index. Thus, when the light passes through the granules, there is a lot of deviation occurs. Phase changes occurs. And in the nucleus, the refractive index is moderate, thus little variation occurs. Okay. So this is called the phase changes in the light rays. How the phase changes in the light rays happens? When it passes through the different refractive index material of the cell on object. So this is a light rays. When it passes through the specimen, that is this one, there will be phase changes in the light rays. Some are deviated lot, deviated light are there, non-deviated lights are there, slightly deviated lights are there. These are the phase changes in the light rays. Now, these phase changes in the light ray cannot be viewed by the human eye. This cannot be detected by the human eye. So, now this phase changes has to be converted to the brightness or the light intensity which can be detected by the human eye. In the phase contrast microscope, these phase changes in the light rays are converting into the brightness, the intensity. Now you can observe the cell like this: a light colored contractile vacuoles, dark regions of granules, and moderately darker region of the contractile vacuoles. So this is the object. Now what are the components of a phase contrast microscope? Before that, let's look at what are the normal components of a bright field microscope or the compound microscope. There will be a light. You can see the contents are here, and this specimen plane. There will be an objective lens and an ocular part is here where the image plane is observed. So here in the phase contrast microscope you can see two additional structures. One is called annular diaphragm and the faceplate. The faceplate and the annular diaphragm are the two important structures in the phase contrast microscope. So the phase contrast microscope is similar to the ordinary compound in its optical components. There will be a light source, condenser system, objective lens system, and ocular lens system. This is the ocular lens system. Here, this is the objective lens, and this is the condenser lens, and this is the light source. The phase contrast microscope differs from the bright field microscope in having, and this is the annular ring, this one, and this is the faceplate. Two structures are there. Now the annular aperture, that is this one, is lie below the condenser lensinte തൊട്ട് താഴെയായിട്ട് കാണപ്പെടുന്നത്, which controls the illumination of the object which is placed here. Object is placed here. So to get contrast, now this is a source of light. Some light are diffracted away, so these lights are blocked by this region, the darker region, which doesn't allow the light to pass through it. But the central region only, the central ring region, the slit which allow the light to pass through it to get a contrasting image, and which forms a cone of light. This is a cone of light. So this annular diaphragm helps to create a narrow hollow cone of light to illuminate the object which is placed here. And this is the second part. This is called the faceplate. This one is the faceplate. It's just two portions, the outer and the inner portion coated with the light retarding material. So that when the light passes through the light retarding material, the light rays, it's closed down. And this is the region where the portion is devoid of retarding materials. The light can easily pass through it, goes through it. There is no delay of the light. So these two regions are very important in the phase contrast microscope. So what is the working mechanism of phase contrast microscopy? So in unstained cells cannot create a contrast under normal microscope. But in a phase contrast microscope, when the light passes through an unstained cell, that is this one, take for example, it encounters regions in the cell with a different refractive indexes and thickness. Correct. Contractile vacuole, granules, and nucleus have low, high, and moderate refractive index. And what happens when the light rays passes through an area of high refractive index? That is this one. This is the high and low, moderate refractive index. The light rays deviate from its normal path. So this is the normal path but deviates because of the high refractive index. Here it is a moderate level of deviations. And this is called the phase retardation. This is known as what? Phase retardation or phase changes occurs. Deviation of light. Now, when the light rays passes through an area of less refractive index, for example, here in this picture, contractile vacuole, when it passes through the contractile vacuoles, it remains non-deviated because of its thickness is or density is lower. The light can easily pass through it without any deviation, so it goes through straight. So finally, so this is the light rays passing through the specimen. We'll get a deviated light rays and a non-deviated light rays are there. So this is called the phase changes. And now this deviation cannot be viewed through the naked eye. This cannot be viewed through the naked eye. And these phase changes in the light rays has to be transformed into brightness. Intensity of light to convert here. So the final image obtained like this. The granules appears are dark in color, the nucleus in moderate dark color, and the contractile vacuole appears light in color. So this is the final image obtained through the phase contrast microscope. And how this is possible? Let's look at it. Said there are two light rays. One is called the retarded or deviated light rays and unretarded light rays are there. And there is a difference. And what is the difference among them? So there is a difference in the phase between the retarded and unretarded light rays is about 1/4 of the original wavelength. Let's look at what is it. So this is a light source. It passes through the annular diaphragm, then through the condenser lens and passes through the object. So this is the specimen. So when the light passes through the specimen, based on the refractive index, it undergoes deviation as light changes in the wavelength. So we'll get a two light rays. So this light ray is unretarded. It goes straight without any deviation, and this is also without any deviation straight. So these are called the unretarded or non-deviated light rays. So when it passes through the object, because of the refractive index, it undergo deviation. So there is a slight retardation. So this is a retarded light. And what is how much it is retarded? Now let's look at is the wavelength. That means how much? So it is represented as lambda. So this is what? Lambda by, this is lambda by 2. This one. This is the lambda by 2. And what is this one? This is this one. This is the lambda by 4 of the wavelength. So this much changes occurs. So as a result, these light rays reaches the faceplate little slower. So these phase changes cannot be detected by the human eyes. So what to do next? Then we use the faceplate which convert these minute phase changes. These are the phase changes, lambda by 4 wavelength changes are there, into brightness or amplitude changes, so that a contrast difference can be created in the final image. So the function of the faceplate is to make a contrast differences between the non-deviated and deviated light rays. So this can be easily detected by the human eye. Now let's Look at how this happened. Now let's look at this structure. This is a non-deviated light, and this is the light passing through the bacterium and get deviated here. So the ray is deviated by 1/4 wavelength, and it is called out of phase because its trough and its peak are in opposite direction, hence it is called out of phase. So when the deviated light having a phase difference of λ4, that is this light passed through the phase retarding material. So this is the phase plate which are having the phase retarding material. Through the current, when it passes through the phase retarding material, this phase retarding material, they experience a further phase difference of λ/4, so that the final difference will be λ/2. The final difference will be λ/2 here. And now the deviated, that is these light rays, and the undeviated light rays, this one, cancel each other out. Now what happens? Let's look at that. So the deviated and undeviated lights are then completely out of phase. Completely out of phase means its peak and trough are in opposite direction, opposite. So this is the peak of one wave, and this is a trough of other wave. This is called out of phase. What happens? They are combined. When they are combined, if these both waves are combined, a negative or destructive interference is obtained. They cancel each other and the amplitude is decreased here, this picture. So this is the wavelength A, wavelength C. So when both are combined together, you will get a decreased wavelength, reduced amplitude wavelength you will get. So this is called a destructive interference. So in that case, when destructive interference, when the amplitude is reduced, you get a darker image against the brighter background. So destructive interference create a darker image against the brighter background. Now, if the undeviated light rays passed through the phase retarding material, the two rays will be in phase, like this, will be in phase. The peak are in phase. So this is called in phase, this is called out of phase. In that cases, the amplitude increases. So this is called constructive interferences. The amplitude brightness increased here. So this is called a constructive interference. So when the constructive interference occurred, the specimen appears brighter against the darker background. So you get two images, darker image against a bright background or a brighter image against a dark background. So both the deviated and non-deviated light has to pass through the phase plate to form the final image. The differences in wavelength, differences in phase, that means the wavelength gives the contrast to clear visibility of the object. The deviated wave produce darker image of the object, while the undeviated waves produce lighter surrounding of the object. So this is the object you viewed under phase contrast microscope. So this is under bright field microscope. So look at the picture differences. So these are the applications of phase contrast microscope. The clear visualization of unstained living cells. It's highly used to, it makes highly transparent objects more visible. It is used to expand various intracellular components of living cells at relatively high resolutions. It helps to studying the cellular events such as cell division. It is helped to visualize all types of cellular movements such as chromosomal and flagellar movement. That's all about phase contrast microscope.